So thanks for the introduction. And as I just mentioned, I'm from the Department of Physics. I'm talking about something which is vaguely biologically related, specifically the extreme swimming and Archaea. And this is something that James alluded to in the last session. I'm interested primarily in developing instruments that physicists can use to investigate biological systems. But the side product of this is that we can use biology to tell us things about physics that we didn't know. I want to start off the talk by describing what the tree of life is. And you've probably seen diagrams that look a little bit like this before. It tells you how everything's related. So at the center of this diagram, there is the last universal common ancestor, the organism from which all of us are evolved from. So bacteria and everything else came from one individual common ancestor. And because biologists, I'm not a biologist, you can tell, I can be rude about them, biologists like to classify things. So the top level classification that people use is what they call a domain. So you can break down the tree of life from this universal ancestor into 3 domains. And they're all represented on this picture. At the top, these are all bacteria, the fantastically diverse lineage there. I'm not particularly interested in those today, although it's an interesting area. The bit I'm interested in is at the bottom, this projection here. And there are 2 main groups, and when I was at school in the textbooks, only one of these groups was there. In the past, they said there were bacteria and there were eukaryotes. And eukaryotes, of course, are the organisms that have their DNA packaged up in a membrane, and that's anything from an amoeba through, cats, dogs, plants, bananas, trees, humans, all of that stuff. All that good stuff is packaged up over here. This group here is the Archaea. And they've only really come into people's consciousness in the last sort of maybe 30 years or so, largely due to the work of this gentleman. This is Carl Woese. He was a microbiologist at the University of Illinois. And in the 1970s, he was investigating a particular type of organism that he thought was a bacterium that lived in swamps. In fact, it's the same things that James was talking about in his talk in the last presentation. And he was using these microbiology tools and molecular biology tools to pull the cells apart to try and find out what made them tick. And he found out that what he was looking at, even though he thought it was a bacterium, the components that they were made of shared some similarity with bacteria, but some of the core components had apparently come from eukaryotes. And then a third section, the components that made up the cells, were completely unique and didn't come from either bacteria or eukaryotes. So we arrive at the conclusion that he'd found a new domain. Not bacteria, they're not eukaryotes. One of the reasons that they haven't really sprung into the public consciousness quite so much as the others is because they live in horrible places. And the name Archaea, the stem from the Greek, is this idea of something that's very old or very primitive. And they live in very horrible parts of the world. Mainly, that's where they're first discovered, because other organisms have come in and displaced them. And in fact, with the new technologies that James was talking about in his talk, we found that, in fact, these organisms are ubiquitous. They live everywhere in the world, but they're most obviously and they're best known in very extreme conditions. So circumstances that are very hot, very cold, very salty, very acidic, this sort of thing. So being a physicist, I went out to go and look for them because I'm interested to see how life solves problems like how do you get around from A to B? How do you maximize your chances of surviving in a horrible world? So this is my physics toy. This is not a £75 million piece of apparatus. This is a $100 microscope that I drilled holes in because it was too heavy to lug around with me. This is a cordless drill battery from a DIY store. This is the Great Salt Lake in Utah, by the way. A laptop at the front there. It's all connected together. It's all battery powered, which is nice because I can put it in the backpack and go around and look for these things. As a physicist in a new field, I'm determined to learn things the hard way. This is the Great Salt Lake in January. The air temperature is -10. The reason the lake hasn't frozen is because it's 3 times saltier than the sea. And my laptop has one of these lovely little touch pads on the front. And I learned the hard way that you cannot use those while using gloves. So you have to take your gloves off. And it was horrible. So we used this microscope, this kind of battery-powered microscope. We discovered some organisms. And thanks to the hard work of a very talented PhD student in my group called Katie Thornton, we established that these microbes are Archaea. So we dragged from a very salty, hostile environment these extremophiles, these things that live at the limits of where we think life is possible. So it's interesting to see how life copes with living in a hostile environment and see what that can tell us about life, not just on the Earth, but potentially beyond as well. So meanwhile, back in the lab in York, this is a much more expensive microscope. This is our sort of favorite technique, if you like. This is a holographic microscope, and it lets us image things in 3 dimensions very fast. You can tell this is a physics lab because we've got all this laser gubbins lying around on the bench, and it illuminates our sample with a light field that's very smooth and even. It's got this unique property of light that's associated with lasers. All the light waves are very nicely in step. And that means that if anything swims across your laser beam, it creates ripples like the ripples in water. So these are the ripples caused by little organisms swimming around in my sample chamber. And by seeing the size of the ripples and where they are, you can follow them around in 3 dimensions and find out how they're swimming. So this is an artist impression. This is not a nice picture from my microscope. This is an artist impression of what a bacterium looks like. So there's the cell body. And because a lot of bacteria need to swim around to find nutrients and get to places where life is better, they've evolved a strategy for swimming which involves helical, rigid propellers at the back connected to a little motor embedded in the membrane. And the motor spins the filament, the cell swims forward. And fascinating, I thought one of the things that really drew me to this in the first place is that it turns out Archaea have the same kind of swimming apparatus, but it's made of different stuff. So bacteria's swimming apparatus evolved from a particular cellular structure in the past. Archaea also have a rigid helical propeller, also have a molecular motor, but it's originated from a different starting point. So life has found 2 different routes to the same engineering solution, if you like, of having a rigid propeller. And it seems that there's something optimal about it, I guess, in some sense, and that's very compelling to me as a physicist. So that's how these cells swim around, but the question is, how do they navigate? I mean, being able to swim is one thing, but you need to respond to stimulus and swim around to find things that are good for you and to swim away from things that are bad for you. So bacteria, under a microscope, this is not a holographic microscope, this is a normal microscope, they look a bit like this. So each dot is a cell of E. coli, as it happens, and they kind of swim around like crazy, little dots swimming backwards and forwards. And the way that they deal with their environment is very random. They're getting jostled constantly by water molecules, and the water molecules are kind of throwing them off course. So the question becomes of how do you navigate in the presence of this randomness? So bacteria tend to swim in a very straight line, change direction, swim in another straight line, and in this way they can sample their environment, and if they sense that life is getting better, they swim a little bit further in that direction. What do Archaea do? No one knows. No one's actually looked at that yet. So there was me in my bucket at the Great Salt Lake, and here's what we found. And I was talking to Katie in the lab, and I was saying, well, is this actually something that's swimming, or is this just silt that's kind of floating around in the sample? How do you know? And the answer is you can look at this, and you can colour each frame in the movie. So if you colour the first frame in the movie red, and the last frame in the movie violet, and all the ones in between, you kind of sweep through the rainbow, and you colour each frame a different colour, you put them all together, you get a picture like this. So the little white dots are just bits of silt sitting there and jiggling around under the influence of Brownian motion. And these rainbow-coloured streaks tell us that there are things in there, but they're swimming very, very slowly. What a funny thing to do. Well, it's living in a nutrient-poor environment, so it makes sense that they want to use energy slowly. The slower you move, the more of the chance that something's going to collide with you rather than you having to look for it. So it makes sense in a way to swim slowly if you're in a nutrient-poor environment. So this is from the Great Salt Lake. We thought, well, this is okay. This is out in the open. There's plant material nearby. The birds are landing in the lake, depositing nutrient sources. This is a horrible environment, but it's not quite horrid enough. So where can we go that's more horrid? And the answer is Whitby. Well, it's a little bit unkind. This is actually Bulby Potash Mine, which is underneath the sea, just off the coast of Whitby. Whitby is lovely. I don't mean to be mean about Whitby. I actually like Whitby very much. So just north of here, there's a particle physics detector facility based in a deep salt mine. And I can show you around. One of the things we're interested in doing is building drones to go sniffing around in these places, to go and collect samples in an automated way. So this is part of the mine. It's a working mine. It's been put aside for scientific experiments. And we're going to fly around one of the tunnels. You'll see the shadow of the drone in a minute. This isn't me just walking around with the thing. We're having a go at flying it around. And so this will hopefully let us get into more hostile environments to pick up samples from places where we don't want to send people. But this is a working lab. There's a particle physics lab. At the other end of this corridor, there's a dark matter particle detector, which is where my wife did her PhD, which is how I found out about this place in the first place. So she's a proper physicist and all this sort of biology stuff, proper physics. So as you can see, the walls in this mine are very dry. Occasionally you find, because it's under the sea, you get seeps of brine coming in through the walls, and the organisms that are brought in with this brine are often very ancient. They've been trapped in the rock for a very long time. There are some studies that claim, whether you believe it or not, that organisms can be trapped in crystals of salt in brine inclusions for 250 million years. I'm a bit sceptical about that. But the experiments are quite careful, and it's kind of food for thought. It makes you think as a physicist, what's the fundamental limit on how long things can live for and stay viable? This seems like a good place to look for these sorts of things. So there you go, drones touchdown. So we collected samples from the Great Salt Lake and from Bulby Potash Mine. We stuck them in our fancy microscope. And we tracked them swimming around in 3D. So we're the first people to do this. One of the reasons is because our flavour of holographic microscopy is unique. There's nowhere in the world who does it quite the same way as we do. Fancy algorithms, lightweight hardware, that's the idea behind it. We can see how these cells swim around. This is about 0.5 millimetre on a side. So we've got a 0.5 cubic millimetre. We can see how the individual cells swim around in 3 dimensions. And what you can see is the trajectories are kind of jiggly. And occasionally, the cells reverse direction. So because these archaea live in very nutrient-poor environments, there's not much competition, the nutrients are sparse, they're playing the long game. They want to swim and stay alive for as long as possible, which makes them a challenge to study in the lab. This is about 60 seconds worth of data. If these things live for a long time, how are you going to study it? This is what physicists do. We can take the parameters that we see from the experiments, how fast they swim, how often they change direction, that sort of thing, we can build a simulation. So instead of watching them for 60 seconds, we can watch them travel meters over a period of days. Right? And we can see, based on the things that we know they do, how they might adapt their strategy to live in these different environments. The conclusion we find is that cells swim very slowly, and we can show that they swim just fast enough to escape the randomizing effect of Brownian motion. So it's just about worth synthesizing flagella and synthesizing this motility apparatus so that they can move, rather than relying on passive Brownian motion to move them around. Moreover, the distance that they run for before they change direction is absolutely critical, and they have tuned that distance or that time specifically so they can harness the randomizing effect of Brownian motion. So whereas E. coli kind of dart around, sampling lots of different locations, the archaea just slowly progress, and they just let Brownian motion carry them off in a new direction rather than having to hare around like crazy. And this is a very low energy, this strategy has a very, very low energy requirement. So that's the kind of punchline of the archaea story. I wanted to mention some other things that we're using this technology for. So the holographic microscopy, I say, is our sort of unique selling point. We're also investigating how human pathogens like leishmania, in a collaboration with Pegeen Walrad in the Department of Biology, we're investigating how these target human immune cells. This is a follow-up on some earlier work on malaria that was done with Sarah Rees in the University of Edinburgh. And the bottom 2 panels, and this side one here, come from a project I'm working on with Martin Bees, who's an applied mathematician, who's looking at how to improve yields in biofuel reactors. So the little dots you can see are algal cells of algae that swim around, and by patterning light and by stimulating them the right way, can we herd the algae to where we want them to be and improve the yield of biological fuels based on their metabolism. So with that, I've come to the end. I'd like to thank some funders, some PhD students who worked very hard on this project, collaborators, particularly Bonnie Baxter and Jamie Butler at the Great Salt Lake Institute, who were very good for filling in with me when I forgot to take Wellington boots on fieldwork, which I learned is the biggest rookie mistake you can possibly make. Also the folk at Bulby Mine and Charles Cookell from the University of Edinburgh, who are great in helping us to get into the mine and do some experiments there. Okay, thank you very much.